Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on module Lipids, Inborn Error of Metabolism, Part 1 from the paper Lipid Metabolism. The objectives of the present module include the understanding of what is actually the inborn errors of metabolism, what are the plausible causes of the inborn errors of metabolism, and how the error can be identified and corrected. If we go back to the history of the inborn errors of metabolism, after the rediscovery of Mendel's experiment in 1900, Sir Archibald Garrett in 1908 was the first to propose about the fact that certain diseases were actually occur due to the inborn error of metabolism. He delivered lectures at the Royal College of Physicians about the four important and major metabolic errors. And those four metabolic errors are alkaptonuria, albinism, cystinuria, and pentosuria. Sir Garrett investigated the cause of the diseases and established the fact that they occur due to some defects in the biochemical reactions of the essential physiological pathways. The name inborn error signifies that the symptoms of the disorders or the defect can be detected in child after birth. Sir Garrett also pointed out that there are certain features in common in all these disorders, like it occurred with a very high frequency among the offspring of consanguineous marriages. The symptoms appeared mostly to early days or weeks of neonatal life, with the familiar occurrence in substantial number of cases with overall benign conditions. The original four metabolic disorders have now been amplified to many disorders. Thus, the dysregulation of the developmental and homeostatic metabolic network, commonly known as inborn errors of metabolism, comprises an important increasing group of genetic disorders. Therefore, it may be defined as an enzymopathy and genotropic disease which is genetically based biochemical problem where an enzyme defect obstructs a defined metabolic pathway accumulating the substrate, causing disastrous pathologic effect at the time of birth. Examples include the phenylketonuria, or it also happens afterwards in life. For example, diabetes mellitus. The four main consequences of the inborn errors of the metabolism are depicted in the following figure. If we look at the concept map, we can see here that the inborn errors of metabolism happens in a metabolic pathway by deficiency of any of the enzyme catalyzing the sequential reactions. For example, here, if a substrate is converting into a product A, which is an intermediate product, and then to a second intermediate product B, and finally to the C product. It involves three steps A, B, C, as well as three enzymes, enzyme A, enzyme B, and enzyme C, and which are responsible for conversion of a substrate to a final product in a sequential reaction. These enzymes results from the three genes also A, B, and C. If any of these enzyme or any gene is missing, it will lead to an accumulation of either the substrate or intermediate product, due to which a disorder happens. This is what exactly happens in case of inborn errors of metabolism. The consequences of the inborn errors of the metabolism. There are four possible consequences of the inborn errors of metabolism. The first is the deficiency of the ultimate product of the metabolism. Example here is the melanin in albinos, which is lacking. The second consequence is the accumulation of an intermediate metabolite. Example here is the accumulation of homogenous acid in alkaptonuria. The third possible consequence is failure of the transport of the abnormal synthesized product. An example here is alpha-1 antitrypsin deficiency. And the fourth possible consequence is the synthesis of an abnormal and less effective end product. Example here is hemoglobin S in the sickle cell anemia. An assortment of symptoms include vomiting, diarrhea, seizures, encephalopathy, hepatomegaly, hypotonia, and failure to thrive. The concept of inborn error signifies the aspect of disturbance in the physiological homeostasis. The metabolic pathway is dependent on anabolic, which is the biosynthetic, and catabolic, which is degradative or oxidative pathway, which are balanced. All the pathway dependent metabolic reactions, whether they are endergonic or exergonic, are catalyzed by enzymes. Metabolic abnormality occurs when there is a defect or absence of one particular enzyme of the pathway as shown in the figure. These metabolic abnormalities are inherited and leads to the most neonatal deaths. In this diagrammatic representation, we can see the effect of the metabolic effect of an enzyme deficiency and what could be the probable consequences of an enzyme deficiency. High level of substrate, low level of product results in an elevation in the substrate concentration, results in the metabolite increased, whereas an elevation in the product results in the metabolite decreased. And these, if the enzyme catalyzing the substrate to product is deficient, then other enzymes modulate the effect of other metabolic activity, as a result of which either there is an activation, inhibition, or competition of the reactions in the metabolic pathway. The significance of these individually rare yet collectively an important disease can be traced back to the importance of biochemical reactions as it is present in approximately 1 in 2000 births and mostly detectable in the childhood. Basically, there are many ways of classifying the metabolic disorders, but when the macromolecules are considered, mainly there are four groups like amino acids, carbohydrates, lipids, and others as depicted in the following figure. In this introductory section, we are primarily concerned with the lipid metabolic disorders. The International Classification of Inborn Errors of Metabolism. The 10th revision of International Statistical Classification of Diseases and Related Health Problems, the ICD-10, contains 22 chapters, of which Chapter 4 contains the information about the endocrine, nutritional, and metabolic diseases. As classified by WHO, the World Health Organization, this system codes these diseases and contains information about the abnormal findings, complaints, external causes of injury, signs, symptoms, and/or diseases, and social circumstances. The fourth chapter contains about 90 subchapters, of which E70 to 90 contains information about the metabolic disorders in the following figures. The codes specifically concerned about the lipid disorders have been depicted. The major codes of ICD-10 involving lipid-based metabolic disorders. The ICD-10 Chapter 4, which explains about the endocrine, nutritional, and metabolic diseases from the subchapters 0 to 90, especially E71, 75, 78, 88, 1 and 2, contains the information related to inborn errors of lipid metabolism. For example, E71 describe the disorders of the branched chain amino acid metabolism and fatty acid metabolism. In E75, disorders of sphingolipid metabolism and other lipid storage disorders have been explained. In E78, the disorders of the lipoprotein metabolism and other lipidemias have been described. Whereas in E88.1, lipodystrophy, which were not elsewhere classified, have been explained. In E88.2, the lipomatosis, which were also not elsewhere classified, have been described. Here the diagrammatic representation presents the major subgroups of the disorders of lipid storage diseases. On the left hand side, which is a part of E75, the disorders of the sphingolipid metabolism, other lipid disorders, GM2 gangliosidosis disease like Sandoff and Tay-Sachs has been mentioned. Similarly, GM2 gangliosidosis, the NOS, which is nitric oxide synthase, adult and juvenile condition, has been described. In E75.1, with classification under other gangliosidosis, NOS, GM1 and GM2 have been described along with mucolipidosis 4. In E75.2, Fabry's, which is Anderson disease, Gawker, Krabbe, Niemann-Pick disease, Farber syndromes, metachromatic leukodystrophy, and sulfatase deficiency have been described. In E75.3, sphingolipidosis, which is an unspecified form, have been described. In 75.4, the neuronal choroid lipofuscinosis disease like Batten, Wyskowski, Jansky, Kufs, and Spielmeyer worked have been described. In E75.5, other lipid storage disorders such as cerebrotendinous cholestasis, Van Bognert, Scherrer, and Epstein-Pullman disease have been described. And in E75.6, the unspecified form of lipid storage disorder have been explained. The disorders of the lipoprotein metabolism and other lipid diseases. Under E78, the disorders of the lipoprotein metabolism, other lipidemias, hypercholesterolemia, familiar hypercholesterolemia, Fredrickson, and other types of hyperlipidemia have been described. In 78.1, again, very low density lipoprotein type hyperlipoproteinemia have been described, along with the Fredrickson and other disorders. In E78.2, where mixed hyperlipidemia, broad or floating, Fredrickson, tuberoxanthoma tuberosum, and tuberoeruptive xanthomas have been described. Whereas in E78.3, hyperchylomicronemia, mixed hyperglyceridemia have been described. In E78.4, other hyperlipidemia, familiar combined hyperlipidemia, and E78.5 hyperlipidemia, which is an unspecified form, have been explained. 78.6, which explain lipoprotein deficiency as well as Tangier diseases. 78.8, other disorders of lipoprotein metabolism, and 78.9 disorder of the lipoprotein metabolism, which is an unspecified form has been described. New developments in inborn errors of metabolism. Actually, the therapy of inborn error of metabolism is progressively more promising, even though expensive. The enzyme replacement therapy emerges to be the safe and effectual for peripheral manifestations in patients with Fabry disease. Gawker disease type 1 and Gawker disease type 3, mucopolysaccharidosis 1, which is also called as Hurler-She and She syndromes, and mucopolysaccharidosis 2, which is also called as Hunter syndrome, mucopolysaccharidosis 6, also known as Marodix-Lamy syndrome, and Pompe disease. Efforts are underway to extend the enzyme replacement alternative for a number of other disorders. Thus, for enzyme replacement therapy has been largely futile in improving the central nervous system. The expression of the lysosomal storage diseases, and repetitively due to the complexity in piercing the blood-brain barrier. This has vanguarded to lively clinical trials assessing the safety and efficacy of the intrathecal enzyme release in numerous lysosomal storage diseases. The accumulated data indicate that for preventing the progression of CNS systems in neuronopathic appearance of lysosomal storage diseases, including some of the lipidose, mucopolysaccharidosis, oligosaccharidosis, sphingolipidosis, and hematopoietic stem cell transplantation may perhaps be efficient under best possible optimal conditions. Although the longitudinal natural history data are limited, published guidelines are available to assist with the decisions related to the pursuit of transplantation and whether to use the bone marrow or the umbilical cord blood derived cells. In general, the transplantation yields the best results when performed untimely in the path of the ailment, that is, in an asymptomatic affected sibling of a child with a lysosomal storage disorder in centers with experience in performing the transplantations to treat the inherited metabolic disorders, and in patients healthy enough to tolerate the conditioning and transplantation regimen. The manifestation of the inborn errors of the metabolism. Although single gene defects typically result in substrate accumulation, the precise causal pathophysiological mechanism that lead to clinical symptoms are not wholly lucid. The allocation of the accruing material connects with the organs that are affected. Cells of the mononuclear phagocyte systems are principally prosperous in lysosomes, and accordingly are recurrently exaggerated by the lysosomal storage diseases. Glia and neurons are most commonly affected, liable because of the relative paucity of cell turnover in the central nervous system. Yet, non-neuronopathic forms of lysosomal storage disorders or the diseases exist. The lysosomal storage diseases may possibly result in a rigorous neurodegenerative phenotypes. The milder, typically later onset or adult onset, phenotypes have been recognized and are generally associated to the degree of enduring enzyme activity. The pathophysiology of inborn errors of metabolism. In general, the accumulation of non-degraded substrate is thought to associate to the cellular dysfunction and death that complements the lysosomal storage diseases. The precise mechanisms underlying this deterioration are incompletely defined. The pattern of neuronal degeneration in subtypes of lysosomal storage diseases may be surprisingly cell type specific. In some cases, substrate accumulation is also associated with sequestration of important component molecules, leading to a relative deficiency state. The lysosome dole out as a vital element of the endosomal lysosomal system. This system is essential for the safeguarding of the typical cellular metabolism, operational in conjunction with the chaperone-mediated autophagy and ubiquitin proteasomal systems. The anomalous function of this system may perhaps lead to ectopic dendritic sprouting, which is a characteristic quite imitable to lysosomal storage disease. And also, it impaired the recycling of glutamatergic AMPA receptors. Neuroaxonal spheroid formation is a feature of the ganglioside storage diseases, Niemann-Pick type A and C, and emanocytosis, implicating a shared pathology that leads to the production of these compact accumulation of mitochondria and tubulovesicular bodies. Now, how to test the inborn errors of metabolism? The constellation of the dysmorphic features like coarse faces, macroglossia, bony abnormalities include dysostosis multiplex, cardiac involvement like arrhythmia or cardiomegaly, in liver like hepatosplenomegaly, ophthalmologic signs such as corneal clouding or macular cherry red spot, and neurological features might guide to the irrefutable distrust of a lysosomal storage disease. Symptoms are typically gradually progressive rather than episodic, as occurs with other neurometabolic disorders. The neurological symptoms can include ataxia, developmental delay, epilepsy, complex partial or myoclonic, hypotonia, intellectual disability, peripheral neuropathy, and or spasticity. The screening can be executed via the skeletal radiography to look for the facts of abdominal ultrasonography to identify hepatosplenomegaly, dysostosis multiplex, which is observed in numerous lysosomal diseases, and echocardiography to assess for the cardiac association. Hearing screening results might be irregular in some cases. However, the ophthalmologic consultation could be useful in recognizing the corneal clouding or cherry red spot. A peripheral blood smear can reveal white blood cell vacuoles, which is an autophagic vacuoles, fingerprint lipid, whorls, granular, zebra bodies that possibly will provide imperative diagnostic clues. Urine can also be examined for the prominent excretion of the oligosaccharides. That is for understanding and looking for the cases related to oligosaccharidosis. And glycosaminoglycans, which happens in mucopolysaccharidosis cases. Blood chitotriosidase, an enzyme which is a marker for the macrophage activation, may have been elevated in case of inborn errors of metabolism. The state-of-art testing is the most proficiently executed by enzymatic activity measurement in a reference laboratory. Characteristically, in peripheral white blood cells, even if skin fibroblasts may also be used. For some disorders, the enzyme activity can be measured in dried filter paper blood spots, and occasionally enzyme activity measurement in other tissues such as muscle can have utility. Urine enzyme activity measurement are seldom helpful, although urine substrate excretion can provide useful information. In some cases, the confirmatory DNA mutation analysis may be an indicator for inborn errors of metabolism. the steps in the therapy for metabolic diseases. There are few steps in metabolic therapy for the inborn errors of metabolism, and that steps include the reduction of the precursor substrate load, provide caloric support, providing fluid support, removing the metabolites via dialysis, diverting the metabolites, and supplementing it with the cofactors. Although the inborn errors of metabolic disorders arises from mostly one enzyme deficiency, yet overall, as all the signaling pathways are interlinked, therefore it behaves as a complex trait. However, other aspects should also be considered, like deficiency of cofactor required for enzymatic activity, or hyperactivity of an enzyme, or problem in transport process leading to an excessive accumulation of the metabolite. While within a signaling pathway, the concept of upstream and downstream metabolite is also crucial, as an accrual of upstream metabolite can divert the pathway of this particular metabolic flux to numerous side pathways. The inborn error metabolism team at the Cochrane Cystic Fibrosis and Genetic Disorders Group declare the 28th February as a Rare Disease Day. This diagrammatic representation explains the different types of the inborn errors of metabolism, like lipid storage diseases or the lipodosis, the fatty acid oxidation disorders, the defects in this cholesterol metabolism, the paroxysmal lipid-based diseases and disorders, and other lipid disorders arises due to the inborn errors of lipid metabolism. So students, today in this lecture, we have discussed about the inborn errors of metabolism, what actually it has, how it is happening, what are the plausible causes, how many types of inborn errors of metabolism have been reported, the manifestation caused by the inborn errors of metabolism, its pathophysiology, and the testing methods that have been employed to know the symptoms of the inborn errors of metabolism. Thank you.